Assumptions of a two-way ANOVA. The assumptions of a two-way ANOVA, they are similar to those of the one-way ANOVA. The assumptions of the two-way ANOVA are similar to those of the one-way ANOVA. One, variable types. We don't always put these in the assumptions, but I want to highlight this here because it's really critical to get this clear in your mind. The dependent variable y must be quantitative continuous, must be quantitative continuous, while the two independent variables, or x's, must be categorical with at least two levels. Two, independence. The observations should be independent between groups and within each group. Independence, it's always in there. Normality. For small samples, data should follow approximately a normal distribution. For large samples, usually n equals or greater to 30 in each group or sample, normality is not required thanks to the central limit theorem. But be careful, because when you're talking about multiple factors, you're crossing them, and you need to have 30 in each crossed group. So it's not just 30 males and 30 females. You need 30 males of Adelie and 30 females of Adelie, 30 males of Gen 2, 30 females of Gen 2, right? Of each. That could mean if you have, in this case, one factor with 2 levels and one factor with 3 levels, you have to multiply those. Okay, now you have 6 groups, 2 times 6, or 2 times 3, right? 6 groups. And you need to have 30 samples in each one of those groups. So 30 times 6 is 180. Now you've gone from needing to take 30 measurements to at least 180 measurements, right? As you add factors, as you add levels, you need to increase your data collection effort substantially, which is why you need to plan this in advance. And statisticians often seem to be telling researchers, oh, wait a minute, how many different levels and how many different factors are you planning to collect? Because you're going to have to collect a lot of data if you want to analyze it with that many different levels and that many different factors. Maybe simplify it, shrink it down, get enough data per category, get a good analysis, and then based on that, maybe do another experiment, right? Next, we need to look at the equality of variances. Variances should be equal across groups, same thing as for the one-way ANOVA. And then outliers, there should be no significant outliers in any group. Right? We're familiar with all these by now, right? Let's check our assumptions. Let's check our assumptions one by one. Variable type. Okay, the dependent variable of body mass is quantitative and continuous. Good. While both independent variables, sex and species, are qualitative or categorical variables with at least 2 levels, it obviously wouldn't make sense if they had just 1 level, because why? What are you comparing, right? Therefore, this assumption is met, right? If your dependent variable is quantitative and discrete, like count data, which is, strictly speaking, should be analyzed using a generalized linear model, not an ANOVA. That's something a little bit different we haven't gotten to yet. So you really want to have that continuous variable. Now, it's a little bit tricky sometimes because you're like, well, we're counting the number of grams. Yeah, okay. Or if you round it to the nearest number of grams, now maybe it looks like a discrete variable. It can be a little bit tricky. Just remember that with a discrete variable can only take on a certain number of values. And so if you think about like test scores, 0 to 100%, right? That's a discrete range. It's not the infinite number line. It's a discrete range. So that would be a discrete variable, even though between 0 and 100 is a pretty big range, and that might end up working pretty well in a two-way ANOVA as a response variable because your range is really big. But now let's think about, let's say your test score can range between 0 and 10. Now that's a really small discrete range, or 0 and 5, a really small discrete range. And that starts to not work as well in an ANOVA. So it's like if you have discrete data, pay attention to that, right? Pay attention, oh, what's my data range? And it starts to look a lot more like a categorical variable when you have a very small range of discrete data. I hope that's helpful. It's not such a hard and fast rule. I do see people doing analyses with discrete data as the response variable, and it's okay. I even see it in textbooks, and it's like, well, but technically that's discrete, but you just ran it. Okay. Because it had a really big range and it acts essentially as a continuous variable. There you go. Let's just get this done. All right. Independence. Independence is usually checked based on the design of the experiment and how data have been collected. To keep it simple, observations are usually independent if each experimental unit, here, a penguin, has been measured only once and the observations are collected from a representative and randomly selected portion of the population. So you randomly selected from this whole population and you did not keep measuring the same penguin over and over again. Like you're like, I measured that penguin, I tagged it, or I marked it, or I'm like, here's a cookie, go over there. I don't know. What do you do? Like, make sure you don't measure the same penguins. Like, a big group of penguins, they all kind of look alike. Don't measure the same one. Like, go over there. Then they're independent. There's other ways that data could be non-independent. Like, it could be non-independent if there is a bias according to spatially correlated data. Like, let's say you went to this island and you only measured the penguins that were like on this one beach of the island, and they could have been on all the other beaches. Then maybe they're not really that randomly selected, and maybe they're not really that independent because they're only the ones that are on this beach, and maybe they vary on this beach in a way that's different from the way the penguins vary on that other beach. There's like some systematic difference, like, oh, this beach has a lot more fish, so they tend to be bigger, or something like that. Yeah, right. Or let's say you could have some non-independence in relation to time. Like, I measured all the penguins in the Arctic or the Antarctic summer, and maybe that's very different than the way they are in the Antarctic winter. And but I don't know how you're going to get there in the wintertime. Maybe you just have to be like, well, that's what we got. We're calling that good. But yeah, there could be some non-independence in the spatial and the temporal realms, as well as measuring the same individual over and over. Again, this is really something you have to think a lot about when you're doing your study design, especially if you're doing something that you'd imagine is very expensive, like going to the Antarctic to measure penguins. I mean, you got to get that fancy boat. You need all those winter coats. It's not easy. Oh, I like penguins, though. They're so cute. That's oil. I'll see if I can find the little penguin video and put it on here too. They may not be independent if each experimental unit equal penguin here, has been measured at least twice, as is often the case in the medical field, for example, with two measurements on the same subjects, one before and one after treatment, which we did before with our pulse lab, jumping jacks pulse before and after. There are ways of taking this into account in the analysis, but we will not cover it here. We will not cover it in this class. It will be, you'll have to learn that somewhere else in another class. In our case, body mass has been measured only once on each penguin and on a representative and random sample of the population. So the independence assumption is met. Our researchers were rad and they did a great job designing this experiment. Note that if your data corresponds to observations made several times on the same experimental units, for example, if one of the factors is a treatment, A or B, and the second factor is time, day 1, day 2, and day 3, and measurements are made at each time point on the same subjects, a two-way mixed ANOVA should be used. That's the other kind of analysis. You can add this other thing of time or space as what's called a random effect. Again, we won't do that. We're going to keep it simple. 3, normality. We have a large sample on all of our subgroups, each combination of the levels of the two factors or each cell, right? Each of the 6 groups. So normality does not need to be checked. It's nice to do, but we can assume it's probably okay because we have a big sample size. Now let's check the sample sizes for each combination of the levels of the two factors. So what we can do here is use this table command, and you basically put in the first factor, species, and the second factor, sex, and you sort of cross the data, and it's going to give us a count that is the number of females of each species and the number of males of each species. And you can see we have more than 30, like the smallest group is 34. And then we have a really balanced design in terms of male and female. We got like pretty much 50-50. But that we do have a lot more samples of the Adelie and a few more Gentoo than the chin strap. There's like, half as many chin straps. Maybe they're a less common species, or we just couldn't find as many for whatever reason. So for completeness, we still show how to verify normality as if we had small samples. I'll show you that just so you know. All right. I feel a sneeze coming on. Sorry, my landlady has a cat that hangs out at this table, and I'm just getting these allergies. Like, all right. So there are several methods to test the normality assumption. The most common being a QQ plot by group on the residuals, and or a histogram by group on the residuals, and or a normality test like a Shapiro-Wilks test, for example, by group or on the residuals. So remember that sometimes we're checking the normality of the data and sometimes of the residuals. So if we want to look at a QQ plot of the residuals, which tells us the normality of the residuals, then we first run the model. It's an iterative process. We have to run the model, check the assumptions, then look at the results, right? Maybe change the model. Okay, so to run the model, we're going to use the AOV command, AOV, which is ANOVA. We did this before. And we always have the Y variable first, and then our tilde, and then we say our first factor, sex, times. We're using the multiplication sign here, say we're times. This is basically the same as saying sex plus species plus sex by species, but it does it all at once in one command. So sex times species, it's going to test those three hypotheses, the main effect of sex, the main effect of species, and the interaction effect all at once. And our data, again, here, make sure you have data equals underscore C, so we're working with the cleaned data. All right. Right, run this, and it's going to save it in something called mod, like modern. No, model. It's the model because this, we call it a model. Now we can draw the QQ plot on the residuals. We show two ways to do so. First with the plot function, and second with the QQ plot function in the car package, which has more than just cars. The whole car package has a lot in it. Plot the residuals using a QQ plot. If the dots fall close to the line, the residuals are normal. So here's method one, plot mod, which equals 2, which is going to say this is the graph we want to look at. Remember, if we just say plot mod, we get 4 plots. We just want to look at this one, QQ residuals. If the dots fall on the line, it's normal. Those dots look pretty close to the line. It's normal. Here's another way of looking at it, which is kind of nice because you're like, well, they're on the line, but like, are they really close enough? I mean, they're kind of a little bit off. Well, here's a way of making that more objective, I think, by drawing the confidence intervals around the line. So you use the car library and you use this QQ plot function on the model residuals. And when you run this, what you get, and again, I just like this a lot because it takes a little bit of the subjectivity of like, how far from the line is far? How close is close? You can see, well, if it's within the shaded blue area within the confidence intervals, then that's close. If it's outside, it's far, right? Does that help you? That helps me. Okay. I like this a lot. I would, if I were you, I would remember this code. I would always save, oh, shoot, I didn't. Let's save this. Save this. I'm going to just put this right now in my biostats folder. I'm going to put this in my biostats folder under this week, right? I know I'm doing Two Way ANOVA this week, and I'm going to save it as Two Way ANOVA Penguins and my name. So I'm not going to overwrite my existing file. I'm going to save it as my name so I know this is what I did, right? With my edits, my answers to the questions. Okay, great. So now we can say, yes, it's normal within those confidence intervals. Super. Okay, so again, if the points follow the straight line called Henry's line, by Henry, I don't know, Henry, and fall within the shaded blue confidence band, we can assume normality. This is the case here. Boom, check. If you prefer to verify normality based on a histogram of the residuals, here's the code. I love histograms. I love histograms. I think that they're just easy to interpret. For me, they are straightforward and visual. If you have a symmetrical bell-shaped curve, it's normal. It's hard to get a more symmetrical bell-shaped curve than this one. So our residuals are very normally distributed, super good. Cool. So now let's go to question 4. Interpret this histogram. What does the shape of the histogram tell us? Does this verify our assumption of normality? Does this support what we saw in the QQ plots above? Remember, I am asking you in your homework and elsewhere to interpret the diagnostic plots. These two diagnostic plots, the histogram and the QQ plot, tell us about the normality of the data, right? So the histogram tells us that the residuals of the model, right? That's what we did this histogram on. We didn't do it on the data. We did it on the residuals of the model data, right? Or of the data from the model. That the residuals of the model are normally distributed because the histogram is, and here's the two keywords that we're looking for, symmetrical and bell-shaped, right? Basically, the shape of the histogram tells you that. Right? Does this verify our assumption of normality? Yes. So yeah, the histogram tells us that the residuals are normally distributed because the histogram is symmetrical and bell-shaped. And does this support what we saw in the QQ plots above? Yes, this supports what we saw in the QQ plots where the points fell close to the line, again indicating normality of the residuals. Gotcha. Okay, good. Note that if the normality assumption is not met, many transformations can be applied on the dependent variable to improve it. The most common ones being the logarithmic or log function in R and the Box-Cox transformations. If the normality assumption is still not met on the transform data, the non-parametric version of the two-way ANOVA, the Scherer-Rey-Haarer test, can be used. Alternatively, a permutation test can also be used. We have options. Homogeneity of variances. Okay, equality of variances, also referred to as homogeneity of variances or homoskedasticity. That's a fun word, homoskedasticity, can be verified visually with the plot function. Okay, so let's look at that. Let's look at that. Now we're going to use plot on our model. Again, that's where we put the results of the model in this thing called mod. So we're going to plot the mod and we're going to say which equals 3. So we're going to take the third plot that comes out of that series of plots we get from plotting the model results. And we can look at the scale location plot. And this tells us about the homoskedasticity, the homogeneity of variances. Remember, the rule of thumb for ANOVA was that the largest standard deviation had to be less than or equal to twice the size of the smallest standard deviation, right? So if you looked at the variance of all those points, right? How much variance they have, and that's what residuals are. So here's the residuals, right? Then you would see, okay, we've got this much variance for one group and this much variance for another group. Oh, that's a big difference in the amount of variance, right? So we've got residuals ranging. So the largest standardized residual is not more than twice the smallest one, right? Here we can see that. Okay, let's break this down. What are these stacks of dots? What are these stacks of dots? So this is the standardized variation, a measure of the variation of each one of these points, right? And you can see that our standardized residuals for each group, so we've got 1, 2, 3, 4, 5, 6. So I'm gonna guess that, so these are our fitted values again. This is what you would predict it to be. Our fitted values for one species, male and female, another species, male and female, and our third species, male and female. Probably that's what it is. This is one species, male, female, right? So we have 6 groups, and this is each group. And you can see their residuals. Those stacks are pretty much the same height. They're roughly the same height, right? Which means that this line across is roughly even, and they're the same height. And the residuals, actually more relevant, is that they're all about on the same line. If you had, let's see, one of these that had much higher residuals, then that stack would be much higher than the other stack. Like all these stacks would be down here, and that other stack would be way up here. And what would that red line do? It would go shooing, right? It would be looking weird, but it's not. It's horizontal. It's roughly flat and horizontal because those stacks are all about the same. So homoscedasticity, same variances. You interpret it by saying the red smooth line is roughly horizontal and flat. That's how you can use these same words for interpreting your diagnostic plot. The red smooth line is roughly horizontal and flat. You can just type that out, which tells us that the spread of the residuals is constant. So it looks like the constant variance assumption is satisfied here. You could use those words for interpreting your diagnostic plots. I'm giving you this as an example to use. Okay, the diagnostic plot above is sufficient, but if you prefer, it can also be tested more formally using the Levene's test, which is also from the CAR package. Now, we don't always do this. Just putting it for you here. If you're someone who really likes to have that test, you want to have a p-value, you want to say yes or no, it's, the assumptions matter not. That's like the Shapiro-Wilks test. You're really testing the assumptions here. You can use this Levene's test. And what you get here is a p-value that is greater than 0.05. You got to be clear on your null hypothesis here. Your null hypothesis is that the variances are equal. There is no difference in the variances, right? The variances are equal. If you don't reject the null hypothesis, then yeah, the variances are equal. So if your p-value is greater than 0.05, you accept the null hypothesis, the variances are equal. Good, you've got homoscedasticity. If your p-value is less than 0.05, you reject the null hypothesis, your variances are not equal. You don't have homoscedasticity. You did not meet your assumptions, and you're going to have to do some transformation or use a different test. Okay? That's the Levene's test in a nutshell. Okay, so in this case, it says right here, we do not reject the null hypothesis that the variances are equal because the p-value is greater than 0.05, p equals 0.227. And here is another way you can use a version of these words in your homework. When I say interpret the diagnostic plots, this is something you could say. Both the visual and formal approaches formal hypothesis testing approaches give the same conclusion. We do not reject the hypothesis of homogeneity of the variances. In other words, the variances are homogeneous. Great. Note that as for the normality, the logarithmic and Box-Cox transformations may improve the homogeneity of the residuals. We did the log transformation last week. We didn't do Box-Cox. It's a similar idea. If you need to look it up, it's easy to do, actually. Lots of different transformations. Outliers. Okay, number 5, outliers. The easiest and most common way to detect outliers is visually, thanks to box plots by groups. Super easy to check this. We've done it loads before. Let's load up our ggplot2 library for our easy and elegant plotting. Library ggplot2. Again, I suppressed the warnings here just so we wouldn't get a bunch of red gobbledygook in our reports. And there's a couple different ways of coding in ggplot. If you want to just see like this way, I like, because it puts everything on a different line with the plus sign. You can see like you're building this layer cake of a plot. So first you say, okay, I'm going to make a ggplot. Hey, R, make me a ggplot. Well, what kind of data do you want to use? Well, I want to use the DAT data. Oh, okay. Well, now I got that. And then plus what? Plus what? What do we need? Well, what's your X and Y? What's your aesthetic? Aesthetic is your X and Y. Well, the X is equal to sex. That's what we want on our X. And our Y is body mass in grams, right? Plus, well, this tells us what kind of a plot we want. The geome, the geometry. We want a box plot. And then just open parentheses. You can add a bunch more. We've done that before, but just running this, we get a neat, very tidy little box plot where we can see we've got male, female, NA. Oops, you guys, I forgot. Okay, would go back up here to that first line of code where it says ggplot. You see where it says DAT? Oops, I forgot. And I was supposed to put DAT underscore C. Otherwise, it gives you this NA as another category and makes a box plot for that. And then it looks like you have 3 categories, which that's not what you want. All right, so now I'm going to change that DAT to DAT underscore C and rerun this. Okay, that's what we want. We want these 2 box plots next to each other for female and male, right? And we can see right away, just looking at these box plots, okay, the dark line is the median. It's not exactly in the center, but it's not like super squished one way or the other. We're looking for outliers here. Outliers would show up as dots on either end of the whisker. I don't see any outliers. So our data is roughly normal and there are no strong outliers, at least for sex. Now we can go look at the box plots by species. And here I remembered to put DATC in, so that should work. And if we run this code, now we can see if we break it up by species. Okay, we do have a couple outliers on our chin strap. On our chin strap, we have a couple outliers, but they're not hugely strong outliers and they're symmetrical, interestingly. We kind of have a really big one and a really small one, but they're not skewing the data strongly from one end to the other. So we do have kind of high variances as a result, but we don't have outliers that are likely to cause a problem with our data. So I'm just going to say there are, as defined by the interquartile range criterion, we talked about that before, 2 outliers for the species chin strap. These points are nonetheless not extreme enough to bias the results.